Okay, hi everyone. Welcome to our first session of Sybeco in 2025. We are excited to have these sessions now recorded, and we will upload them to our YouTube channel. Sybeco is this initiative that we started like 2 years ago at Georgia Tech, and before that I did that for 2 years during COVID at the Allen Institute, not under the name Sybeco, but the idea was that I would invite the first or second authors, the lead authors of papers who are always trainees to present their work. And the idea was to both help trainees to get recognition and be known by the institutes, research institutes, and also help with networking and collaborations down the road. So today we're very excited to have Hugo Ninou from Yifat Prut Lab, and I will let him introduce himself more with the bio. Yeah, go ahead. Thank you, Hugo, for joining us.
Thank you for inviting me. Hi, everyone. So yeah, I'm Hugo Ninou. I'm a French student from ENS in Paris. And in 2022, I did an internship in the Hebrew University in Jerusalem, mostly under Jonathan Kadmon supervision. But this is work that was done conjointly with Yifat Prut's lab, who is actually responsible for most of the data, the acquisition of most of the data that you're going to see today. So I'm here to present our latest work on how cerebellar output shapes cortical preparatory activity during motor adaptation. So to get a brief understanding of what I mean by motor adaptation, I want to take a small example of a monkey that you can see here that, let's say, wants to learn how to ride a bicycle. And this is going to take a whole lot of training, motor coordination, and equilibrium. And essentially, after the monkey has learned how to perform the task quite well, it may want to take a ride down a winding road or in another environment, such as from the top of a mountain, for instance. So hopefully, the monkey is not going to have to learn how to ride a bicycle from scratch in each of these new environments, and is going to be able to adapt what it has previously learned to these two new environments. So this is what I call motor adaptation, and what's going to be referred to as adaptation in this work. And so this is a quite remarkable ability of the brain to be able to adapt to new environments. And there is, in particular, one pathway that has been identified for being responsible for it. And this is the cortico-cerebellar pathway. So as you can see, there are quite a lot of connections over there, but I'm going to try and make it simple and run you through this cortico-cerebellar loop. So I'm not sure if you can see my pointer on the screen.
We can, yeah.
All right, so the motor plan originates in the motor cortices over there. And then the motor plan is going to be transmitted to the motor neurons down the spinal cord. And in the meantime, there is an efferent copy of this motor plan that is also going to be sent to the cerebellum, to the cerebellar cortex. So the cerebellar cortex is going to take both information about this efferent copy of the motor plan and also proprioceptive feedback from sensory areas. And essentially, I mean, it's been proven that the cerebellum kind of operates a comparison of these two signals, and which then allows for the refinement of the motor plan in the motor cortices. So thanks to this comparison of proprioceptive feedback and the efferent copy of the motor plan, the cerebellum is able to send kind of an update information through the thalamus down to the motor cortices for it to enable live adaptation live refinement to the motor plan. So this is this pathway that's highlighted in green over there, and this is the main subsection of the pathway that we're going to be interested in during this talk. So there is another point that I'd like to introduce before diving into the paper result, which is the fact that this motor plan actually happens already way before the movement starts. So the cortical activity goes essentially in two phases, one preparatory phase and one movement phase. And essentially, one remarkable property of these two phases is that they happen in orthogonal dimensions to in orthogonal subspace in subspaces that are orthogonal to one another. So here in this vertical axis, you can see the what is represented as the null space preparatory dimension that is sometimes also being called the output null subspace, in that it doesn't trigger motor neurons. So this allows for the preparation to happen while no motor command is being sent down the spinal cord. And then there is going to be, at the moment of the beginning of the movement, translation to the execution subspace or the readout dimension, which is the movement subspace. Right? So this translation is represented over there, and it happens in a dimension that is orthogonal again to both of the execution preparatory subspaces. All right. So we have these two elements, the fact that the preparatory activity in the cortices happen in two phases which are orthogonal to one another, and the fact that cerebellum plays a role in particular during the movement phase that has been identified before. And so this raises the following question, which is, well, is there any role for the cerebellum already during the preparatory phase? So this is the question that our paper tries to answer. And so in order to do this, Yifat Prut's lab has developed a method which essentially consists in blocking the cerebellar signals that map onto the cortex. So this is what is going to be referred to later on as high-frequency stimulation. And essentially it consists in stimulating the peduncle that maps from the cerebellum to the thalamus, and then, therefore blocking the information outflow from the cerebellum to the cortices, to the motor cortices. So this is a paper from 2019 from Nashev et al. From in Yifat Prut lab. And what we are going to do is to perform this procedure while recording the activity during the motor cortices, and while having the monkey perform these tasks that require adaptation. So the usual experimental paradigm for studying adaptation in monkeys is the center outreaching task. So here is how it goes. So during this pre-Q period, the monkey is going to have to hold the controller and hold it in a central position. After which the monkey is going to be shown a cue, that means a target that the monkey is going to have to reach later on. During this delay period, the monkey is going to have to remain in the central position while preparing for the forthcoming movement. And then a second signal is going to be sent to the monkey that indicates that the monkey can now go and reach for the target. So as we're looking, we're interested essentially in the preparatory activity. The time period that we're going to be interested in is going to be this delay period. And here you can see, so these are hand trajectories of the monkey in different conditions. So you have the control condition where the monkey has, when the cerebellar signals are fully available to the motor cortices. This is what happens during HFS. So when the when the cerebellar signal are blocked. And then we have these two other conditions, which are the with FF denoting the force field. So which is a new condition that the monkey is going to have to adapt to. So essentially, we're going to apply a force field to the controller the monkey is holding. And then there is a condition that combines both HFS and force field, which we call force field HFS. So here is what a session looks like. Essentially, we're going to have this, well, first a control block where the monkey has to perform a few trials under this control condition. Then a force field, HFS, and force field HFS blocks.
Question, Hugo. So HFS, you said, is high frequency stimulation?
Yes.
And what area, where are you applying it? Sorry if I missed that.
Yeah. So the HFS is applied at the entrance of the thalamus on the fibers that come from the cerebellum. And essentially, what has been shown in previous work from Yifat Prut's lab is that this kind of reproduces the symptoms experienced by cerebellar patients, right? So by this I mean ataxia, dysmetria, so this kind of difficulties that people with cerebellar dysfunction experience.
And how do you, because this is monkey, I'm curious, how do you identify these specific cerebellar thalamic fibers?
So, I mean, I'm not an experimentalist, so I wouldn't be able to answer this precisely. Yeah, I'm sorry about that.
Don't worry. Yeah, okay, thanks.
All right, so going back to our blogs, which are roughly made of about 40 trials each, and then they are interspaced by these washout blocks to wash out the effect of the previous block. All right, so if we put things into a table, this is essentially what the four conditions that we are going to apply are going to look like. And something that I would like to highlight is the fact that in the case where we do not have the force field, we're able to record both blocks during which only one target is being presented to the monkey, and during which one out of the 8 targets is presented randomly within one block to the monkey, while during force field, because we're looking at adaptation, we're always going to have one target being presented only at the monkey. Right, so essentially we're not going to have 8 targets for neural recordings within these force field and force field HFS blocks.
Question, Hugo. I guess it would help to give a little bit more information on the behavior. So on each trial, they will have to make a reach to any of these 8 targets, depending on which target is shown to the monkey, correct?
Yes.
And then when they enter these force field blocks, those are the ones that there is perturbation.
Exactly, yeah.
And you're saying that those happen only, the entire block is only one target.
Yes, correct.
Right. Okay.
So one session here corresponds to 1 day. Because the recordings are being done with single electrodes, this means that the neural recordings being performed during 1 day will be associated to one given target, while on another day it will be another. However, for each of these days, we also have access to minus HFS and plus HFS without force field for all of the targets in these blocks. So I would like first to focus on what the neural activity looks like during these control and HFS conditions without force field with these 8 targets recordings. So this is the neural trajectories in the preparatory phase. Essentially, what we can do is that we can stack the activity recorded for each of the neurons and for each of the targets in the fashion that is described over there. So this makes up a matrix that we can then perform a principal component analysis or a PCA on. And essentially, I'm only going to take these 300 milliseconds of the trial that were right before the go signals. So these are represented in these thick lines over there. And this is the data that we're going to use to perform the principal component analysis. And if we look at the first two principal components, one thing that is quite striking is that we are going to see that the neural activity is going to be arranged in the same fashion as the targets are in the physical space, where the neural activity associated to target 1 and 2 are going to be close to one another, and so on and so forth. So they are arranged in this kind of circular way. So this, again, is going to be essentially the preparatory phase, so the null space preparatory dimension that we saw in the previous illustration. And then if we look at other principal components, we're going to essentially be able to identify these translation trajectories, which we also saw previously. So one thing that could be quite interesting is to actually study how these task-related manifold change when we now apply high-frequency stimulation to the monkey. And
question, could you go back, Hugo, please? So just to be sure, the recordings are from the primary motor cortex, correct?
Yes.
Okay, and then the PC plot on the left, that's for the, okay, both of them are for the preparatory phase. We are not looking at the execution yet. All right, okay.
So here, I mean, I forgot to mention this, but these circles with the black outline are going to be the Q signals. So, I mean, the activity at the Q signal, and the full dots are going to correspond to the activity at the go signal. So here the small lines is actually a projection of the activity that occurred before the activity used for the PCA. And what we're going to see is that at the Q signal, the trajectories associated to different targets are going to diverge from one another to go into this circular shape. And yeah, so here we stop at the go signal, so at the end of the preparatory phase.
And then what is the thicker? The line gets thicker.
So the thicker line represents the data that was used to compute the principal components.
I see. Okay, thanks.
Okay, so now we can do the same thing when also adding the data acquired for high-frequency stimulation, which is represented in dashed lines over there. So again, we apply this high-frequency stimulation from the peduncle that maps from the cerebellum to the thalamus, effectively blocking the cerebellar signal to the motor cortices. And we're going to look at how this affects the task representation within the motor cortex. So this is what you can see on the three first principal components. Essentially, we're going to have a global shift of the neural activity due to this HFS, which is going to be target independent. By that, I mean that it's going to be the same shift for each of the targets. So this is what we can see here on this first panel. And if we look at the top view of this representation over there, we're going to see that essentially the two manifolds for control and HFS are going to be superimposed to align on top of one another, roughly speaking. So this is what we can see, that here the dashed yellow line kind of aligns with the full one, and so on and so forth for the all of the 8 targets. But quite interestingly, if we dive a little bit deeper into the principal components, and we look, for instance, at the 5th principal component, we can also recover principal components that show target-dependent effect of high-frequency stimulation on the neural trajectories. So by looking at the principal component 2 over there, we had the two manifolds essentially where he experienced a global shift, while for this PC5 over there, we see that the two manifolds are kind of intertwined, and we have this target-dependent effect on the neural trajectories. So here you have the manifold with the dashed line that goes like this, and the one with the full line that goes like that. So along this PC5, essentially the activity of targets 6, 7, 8 is going to be close to that of the targets 6, 7, 8 during HFS is going to be close of the activity of the targets 2, 3, 4 during control, and vice versa.
All right. Sorry, can I ask some question about the intuition of how to interpret these PCs in terms of firing rate? So if you have a global shift, say for PC2, yes. What does that mean for the firing rate of those neurons?
So this is not. So yeah, we looked at whether this corresponded to a general decrease or increase of the firing rates of these neurons, and actually we find that there is no significance in terms of whether this effect is actually a reflection of general decrease of activity for the neurons. So this means that for some neurons there is going to be an increase of activity, and for others a decrease, and so on.
Okay, can one sort of extract from PC2 what the associated change in firing rate is that creates PC2?
Yeah, so you can extract it by looking at the component. I mean, essentially every component of the PC2 is going to be the change of firing rate for each of the neurons that make PC2. And what we're going to see is that on average, this is going to be 0. Does it make sense?
Yeah, I sort of want to kind of get the temporal dynamics of the firing rate that is associated with the PC2, but never mind. Okay.
So, I mean, here we're not looking at, I mean, so again, so here the representation changed a little bit. As you can see, the thick lines are quite centered and restrained around the go signal with a 50 millisecond time window. So here we're more looking at neural states than neural trajectories or dynamics. But at this given state, the change of neural activity associated to PC2 cannot be associated to a general or global increase or decrease of firing rate for all of the neurons involved.
Okay, thanks.
All right, so ideally, one thing we would like to have would be the same kind of representation, but this time when the monkey is subject to force field adaptation. Unfortunately, as I mentioned before, in the force field adaptation case, we only have access to recordings which are associated to an adaptation to one given target at a time. So we're going to have to do things we are not going to be able to compare these task-related manifolds directly. All right, so if we look at the neural activity for one given target in all of these 4 conditions over there, this is the representation we're going to get in the first 3 principal components, both during preparation and during movement. And essentially, one thing that you can already see here within this 3D representation of the neural activity during preparation is that the cloud of points, each of these points being actually one trial for force field and for null field, are actually closer together during the control condition, so for the blue points, than they are for the HFS condition. So this means that the preparatory activity seems to be more sensitive to force field perturbation while cerebellar signals are being blocked than when they are available. So this is an effect that is actually more pronounced during preparation than it is during movement.
Question, Hugo.
Yes.
So for the force field, which part of the is it after they correct for the perturbation that you're using in your PC or before? So which time points are you using in your PC analysis?
So here, this is average activity during the preparation. So it's I think it's a 300 millisecond period that ends 100 milliseconds before the go signal.
Right. So then I guess that makes sense that you see more of a separation in the HFS condition, because that means that they did not correct for it, right? Because if they had corrected for the movement, then because you're taking mostly these last time points, which is perhaps when the monkey is correcting for the perturbation. So if they're correcting, then force field and null should be more similar. And that's what we see in control. But if they are not correcting, then they are farther. And that's what we see in HFS. Am I seeing this right? Also, would be very helpful to see some behavioral figures. Do you have any? So we see what HFS actually does to the behavior.
So I don't have, so the only behavioral data I have for this presentation is essentially this one, where we can see roughly like the behavior during movement. But here, all of the data we're going to look at is actually data that was acquired during the delay period. So the monkey has not moved already.
Right. Okay, thanks for reminding me of that. And here, the top circle is the center and the bottom is the target, or the other way around?
So the bottom is the center and the top is the target. So we go like this.
I see.
So maybe something that is that is different from other studies in a center outreaching task is that you may notice that actually these circles are quite large. And this was done essentially because the monkey had difficulties performing the task during force field HFS. It turns out to be quite hard for the monkey to do that. So in that case, having larger circles for the monkey was quite helpful.
Right. And then the color code here is for the trials.
Yeah, so that's for the trial numbers, yes.
Okay, which we can see the adaptation, especially in the control form. Right. And then the key here is that because you're doing this perturbation in a block, then the monkey starts predicting that there will be a perturbation. So that's why we see that during the delay period, delay, these trajectories are already, the population trajectories are already different.
Exactly. So as the monkey is adapting to this perturbation, during the delay period, actually, I would expect to see more difference in the preparatory activity the more the monkey is adapting to the condition.
Yes, but then why was it that your null had less difference compared to the HFS? Because if you go to your PC plot,
yes.
So I'm a bit confused then by that, because if there is more adaptation in the null condition, then I would expect the null and force field to be farther in the PC space. But your HFS is showing, yeah.
Yeah. So again, this is the activity recorded in the motor cortices, right? So this essentially you could interpret this as during HFS. So while the monkey does not have access to cerebellar feedback, the monkey is adapting more in terms of the motor cortex preparatory activity.
So when the cerebellum is removed from the picture, they're adapting more?
The adaptation that occurs in the motor cortex is more pronounced.
Is that not a bit weird? Because we always think that the cerebellum drives the adaptation.
I agree. So you could see it in a way that when the cerebellum is there, then the cerebellum does the job and the motor cortex doesn't have to. while when it's not there, then the motor cortex has to do it best as it can.
Interesting. Okay, let's keep going.
So one thing that I should also mention is that the trials that were kept here are the trials that were successful. So the monkey succeeded in adapting to the new condition. So this is an illustration, but in order to quantify that, what we did is that we divided these sets of points into a training and testing data sets, and that we trained decoders to discriminate between null field and force field, both in control and HFS. And what we saw is that during the delay period, so this is in the middle of the delay period, and this is right at the very end of the delay period, the monkey was, in the neural activity, was way more separable during HFS than it was under control. So this means that the neural activity between null field and force field was way more different during HFS than it was during control. So what I would like to...
Yeah? Sorry, is this for, I think I maybe, so is this now for, okay, so, right, so when cerebellar output is removed, then that's when you see that, but, okay, so the decoder accuracy is better when we have when the cerebellar output is removed, which is what you basically showed there in your PC plot, right?
That's it.
And your interpretation is that this is because these are successful trials. So if the cerebellum is not doing the job, then...
So one way to see it also is that we're looking at what happens during preparation. And we could say that essentially the monkey, when subject to force field and has access to cerebellar signal, doesn't have to prepare in a very different way when it has access to cerebellar signals, because it's going to do its movement and then it's going to be able to adapt live during the movement. So the preparation doesn't really need to be so different from the control condition, because the job is going to be done live by the cerebellum.
Very interesting, right?
That's one way to think about it. Okay. All right. So now I'm going to present an indirect way of mapping these single target recordings to the target-related plane that we saw in the representation before. So the way I'm going to do it is by taking this 8 targets preparatory activity that was recorded during preparation, during control condition. So this is the data represented along its PCs as we saw before. And I'm going to project this data, the first few pieces of this data, on linear regression axes that correspond to the mapping of neural activity to the target position. So we're going to map neural activity that has dimension number of neurons recorded to the target position that is actually the X and Y position of the targets being cued to the monkey. So we're essentially going to try and find these regression coefficients so as to predict the target position based on the neural activity. So by projecting the PCs of this data over there on the linear regression axes that are here, we're going to be able to design a plane with 2 new axes denoted here TDR1 and TDR2. And that essentially are going to be, by projecting the data, the neural data on this plane, we're going to be able to have a prediction of what was the cued target in the control condition. So here the big circles correspond to the neural activity of one given target during the control condition. And here I'm going to be able to project single trials from one target conditions onto this plane, which are being represented by the small dots over there. So here, for instance, we could imagine that this is the force field adaptation trials for target number 8. So by doing this, I'm going to find a way to then compare the force field adaptation trials to this target-related plane that have information about the target position that the monkey is aiming at. So essentially what I'm going to look at is this angular deviation alpha, that is the angle between the control condition target and the trials for this one target conditions. So I'm going to be able to show distribution of these angles alpha under different conditions. And so this is what's represented here. So this is in the minus HFS. So in the case where cerebellar signals are available for the monkey under different conditions. So in the case where there is no force field, we can see that here overall this distribution is centered around 0. It's going to be actually the same whether the force field is going counterclockwise or clockwise. So it's going to be a little bit broader distributions in the case of force field adaptation, but overall they are centered around 0. And something quite striking happens when you now remove the cerebellar feedback to the motor cortices. And so this is what you can see here in this plot below. So essentially, if you have a force field that goes in the clockwise direction, well, we're going to see an angular shift in the counterclockwise direction. And vice versa, in the case of force field going in the counterclockwise direction, we're going to see an angular shift in the clockwise direction. And in the case where we have no force field and only HFS, we have this kind of weird distribution that is a little bit bimodal, but that on average is quite centered around 0. So this is the data that we have for one of the 2 monkeys that were used in these experiments. And if I report the average position of each of these distributions on the graph, this is what I'm going to get for monkey S over here. So essentially, we're going to have all of the arrows that point onto 0, and these 2 distributions that point that direction that point in angles that are roughly the inter-target spacing between the inter-target spacing. And so what this hints us is that actually the changes in cortical activity within the target-related plane may drive the residual adaptation under HFS. So everything happens as if when the monkey doesn't have access to cerebellar signal, it's going to adapt by aiming sideways so as to compensate for the force field. Right.
Okay, this is great. I want to make sure I fully understand what's going on here. So here we are seeing where the target position, if it is only coming from the activity of the motor cortex neurons. So that's the idea here. And then we compute the error in the position that is coming, that is predicted from the activity of motor cortex neurons. And you're saying that overall, when there is no cerebellar inactivation, even though there is some error, but on average, it's centered on 0, which means we can predict the target position well from motor cortex activity.
Mm-hmm.
But now when we take the cerebellum out, we see that motor cortex activity predicts a target position that is, for the clockwise perturbation, is counterclockwise.
Correct.
Meaning that if there is no cerebellum, the motor cortex will overshoot, over-adapt. Because we perturbed it clockwise, so the monkey has to do counterclockwise. But the monkey does it too much when we take out the cerebellum.
Well, it's not, I mean, I don't know what you mean by doing it too much, because in the end, the monkey, I mean, has successfully done the trial.
Yes, well, the behavior is right. However, the prediction that we get for the behavior from motor cortex activity will predict.
That's it.
Over-adaptation.
And importantly, here the angle we're measuring is between the activity, the big circle, that of the control condition with other conditions, which are the small dots over there. So we see that actually the neural activity in these conditions compared to the control one is shifted.
Right, but then the fact that the behavior in HFS, you get good behavior. I mean, there was no quantification of the behavior, but it seems like, and it seemed to be more variable in those trajectories that you showed us. But overall, do you have a quantification of the error in reaching the target between HFS and non-HFS conditions?
You mean a behavioral measure of this error?
Yeah. The behavior, the pure behavior, not behavior predicted from neural activity.
Yeah. So the pure behavior, you will see in some error that will start quite high, and then that roughly within 4 to 5 trails reaches a correct value. And this is a little bit slower for HFS than it is for control. But yeah. So these,
and then the trials that are used here are all trials.
Yes.
So you're saying it's a little bit slower. So cerebellar inactivation doesn't do much to the behavior except for slowing down the adaptation. But it does quite a bit to the motor cortex activity.
It does. Well, I mean, it does change the behavior in a significant way. But the monkey is still able to perform the task. Yeah. And then for the motor cortex activity, the neural population activity that we show here, we see that comparatively to the control condition, essentially, it seems like the motor cortex is aiming sideways so as to compensate for the force field.
Sorry, can you see the aiming sideways in the actual trajectory, that the initial part of the movement is sideways and then it drifts back to the target based on the force field? You'd expect, if this is the intention of the monkey, that the initial trajectory would actually shoot out that direction when the animal starts moving.
Yeah. So, so I think this analysis has been done, and actually it was asked during the review rounds when we submitted our article. I don't have the plots here, but essentially yes, you can see this effect indeed in the behavior.
Cool.
At the very beginning.
So this is the TDR, your targeted dimensionality reduction was done on the preparatory activity?
Yes, it is.
So then I guess what we are learning is that when we take out the cerebellum, there is some compensatory mechanism that tries to do this adaptation early on before.
Correct. So this, I mean, the thing with the behavior is that you're already in the movement phase, right? So yes, the monkey is going to have to aim sideways. I mean, this you can see. But here, the interesting thing is that you already see this during the preparatory activity.
Yes, and the sideways is the opposite to the perturbation side. Am I seeing this right? Because they kind of are already trying to compensate for it.
Correct.
This is very interesting.
Yeah, I think it's similar to the dart throwing with a prism on your head. You have the implicit motor learning that's sort of automatic, and you don't have to think about it, and you're correcting for prism adaptation if the cerebellum is intact. If the cerebellum is not intact, the automatic adaptation goes away, but you can force about thinking about how to compensate and pretty much cortically just aim to a different location with a dart, and then you will actually hit the right place. I think it seems like they're just preparing to aim for a new place, and then the force will drift it back, the movement back to where it needs to go, and that then they're all set with the reward. So it's quite cool actually.
And you're saying that this explicit cortical adaptation happens kind of before even the trial starts, but when they go to that implicit automatic cerebellar adaptation, they just wait, and when it happens kind of in a reflex way, they compensate for it. So that's why we don't see much during the preparatory activity if the cerebellum is there. When I say we don't see much, I mean much difference between forceful and control. Yeah.
So
and have you done all? Sorry, go ahead.
So at least in this target-related plane, right, when we are trying to decode the target or the target angle, we see very little difference between the two conditions.
Between which conditions?
Between force field and null field conditions.
Right. Right. And I had one more question. Have you done all of this analysis using not the preparatory phase, but the execution phase?
So I did not. The main a priori issue that I would have when doing this for the movement period is that actually we have a lot of the behavioral activity that would be repercuted on the neural activity that you record. And given that the behavior is different under control and HFS, it would be hard to see what is the difference. I mean, what is due to HFS on the behavior and what is due to what is a direct repercussion of the behavior and what is not, essentially.
Awesome.
So that's why I think it's. I mean, it may be possible, but it's a quite challenging thing to do.
Yep. Thank you.
All right. So, well, actually, I had this intermediary recap, but I think most of the ideas were already raised during this discussion. Well, briefly, so we saw how the neural trajectories during the preparatory phase were affected by having HFS. So with these global shift, so target-independent effect and target-dependent effects, we see that there is some shared representation between control and HFN, and this is actually what enables us to perform this TDR, this comparison of the angles. So we could imagine that this mostly happens in this subspace over there. We also saw that the preparatory activity is more sensitive to force field perturbation during HFS than it is during control. And finally, we saw that the changes in cortical activity within the target-related plane may drive residual adaptation under HFS. So by having only this piece of information, we could sum it up in the following sentence. So preparatory activity is more sensitive to force field perturbation during HFS. And this showing clear changes in the target-related plane, likely compensating for the force field. So one way to interpret this, as we mentioned before, is that this may occur because the cerebellar signals enable live adaptation, and therefore reducing the need for adaptation already during preparation. So given this, one may say that, I mean, it could well be that the cerebellum only influences movement execution, and that it doesn't actually affect cortical preparatory activity, but this is more of a consequence of having to anticipate for the lack of cerebellum that drive these changes. So what I'm going to show in the next few slides is that actually this last statement is not correct, and that we actually, in fact, see that HFS induces additional changes in the cortical preparatory activity. So I'm going to go back to the cases where we do not have adaptation, where we have these 8 target data for both control and HFS. And what we're going to try and do is to look at the dimensionality of both of the task-related manifolds that we saw before. So again, quick reminder on how PCA was performed in this study. So essentially, we have this matrix with all of the data that we can call X. And the way to perform PCA is that to look at the correlation matrix of this data and then extract the eigenvectors and eigenvalues from this correlation matrix. So the eigenvectors are called the principal components and lambda I, the eigenvalues. So this is this leads to this kind of representation when we project the data on each of these principal components. And this is the explained variance, or which is actually the value of the eigenvalues over there. And so we would like to have a dimensionality measure for each of these manifolds that we separately for control and HFS. And in order to do that, we need to build a dimensionality measure, which is this quantity over there. So essentially, we're going to have the sum of the eigenvalues squared over the sum of the square of the eigenvalues. So importantly, this quantity on top is always going to be equal to 1, as if we sum the explained variance for all of the eigenvalues, this is going to sum up to 1. So 1 to the square is going to be 1. And we're only going to be left with this part below. And to give you a rough idea of how this works, I've written down here 3 examples. So if we imagine that we have the first eigenvalue that explains all of the variance and all of the other eigenvalues are equal to 0, then essentially we're going to have 1 over the sum of lambda I squared, which is going to be 1, so 1 over 1. And this dimensionality measure is going to be to give us d equal 1. So this means that the data that we have essentially arranges along a line, which is of dimension 1. Well, if we now take the example that the explained variance is equally shared between the two first principal components, so where lambda 1 equals 1/2 and lambda 2 equals 1/2, and the rest is equal to 0, we're going to still have 1 on top of that over 1/4 plus 1/4, so it's going to be 1/2, which we invert, so it's going to give us d equal 2. So this means that the explained variance is shared equally between two principal components, that the data arranges in a plane. This is dimension 2. So if you take the extreme case where now each of the eigenvalues equally explain a part of the variance, we are going to get d equal the number of cells that we used initially. So of course, I mean, in reality, it's not going to be 1/2 and integers over there, but we're going to have all type of intermediary possibilities. And so this is a real example of what we get for the explained variance for each of the principal components. So if we do it separately for control and HFS, we're going to be able to compare the dimensionality of these manifolds, both in the case of when cerebellar signals are available or not. So this is the results that we get. So this is the measurement done at different points in the time of the trial. Beware here, the axis starts at 5, right? So this is what happens right at the moment of the Q signal. So there is no reason why there would be a difference in these representation between control and HFS. And actually, they are quite similar. But quite interestingly, it's that during the preparation period, the dimensionality of the task representation is higher for HFS than it is for control. And then after the movement starts, well, we recover quite the same dimensionality for both. So here the monkey is not adapting to anything. It's just a pure control and HFS conditions. And this is quite counterintuitive, as what the HFS procedure consists in is essentially sending very structured signals to the peduncle that maps from the cerebellum to the thalamus, right? We're sending to all of the fibers the same high frequency signal. So it's a very close to dimension 1 signal that is being sent to the thalamus, while in control condition, well, it's probably something that's more complex and refined with higher dimensions. So what we see is that by removing these complex signals that come from the cerebellum to the cortex, the dimensionality of the task-relevant manifold actually increases in the motor cortices. So if you want to have a kind of sketch representing what I'm saying here, this would be the task-related manifold in control condition. And this would be the perturbed manifold when the cerebellar signals are being blocked. So the main takeaway from this slide is that actually cerebellar signals shape cortical preparatory activity, essentially by reducing its dimension.
Okay, Hugo. So first of all, beautiful. Thanks for walking us through all of this dimensionality measure and everything. So I guess this though aligns with what you showed us with the TDR analysis and the decoding, because when the cerebellum is not in the picture, the cortex is taking care of this basically beforehand preparation. There's kind of like this, we don't do this online correction anymore. Before that, the cortex is getting prepared. So then that makes sense that the dimensionality will be higher because there is a lot going on. But when the cerebellum is there, not much is going on because online the cerebellum will correct for it. So why would the brain allocate resources during preparatory activity while it doesn't really need it?
Anyway, so that's one way to see it.
But I guess your interpretation was different. You see it as the cerebellum actually quenching variability. So
so so far with the data that we have and the evidence that we have, it's not possible to distinguish between these two hypotheses, right? But in one way, I mean, still it's the fact that the cerebellar signals are here that you see this. I mean, either you see it as the cerebellum quenches the dimensionality, or the lack of cerebellum expands the dimensionality. Essentially, I mean, I think it's two sides of the same coin.
I guess the main way to know that is to record from these neurons during the preparatory activity. If they're not doing anything, then I don't see how that can reduce variability. But if they're active, then that's more aligned with this idea that you have here, that the cerebellar activity is reducing the dimension, because it's doing something. It's reducing the variability during the. So I'm saying that I guess for that we just need to record from these neurons and see. And the other thing is that you're doing also high frequency stimulation, which is, it's not inactivating those, we are stimulating them, which is different from like if we were doing optogenetics inhibition.
Correct.
Yeah, and I can see why this was a surprise, because you would imagine you're introducing noise here. Why would then the dimensionality? Well, sorry.
So if you were injecting noise, then the dimension would
go high.
So this is something that we also checked.
Yes.
Actually, if you were to take the control data and inject noise to it, the amount of noise needed to get to the HFS levels is roughly twice of that is observed in the control condition. While actually the amount of, the amount of noise in control and HFS condition is roughly the same when we measure the Fano factor.
You mean in motor cortex activity?
Motor cortex, yeah. So you see some noise in the behavior. However, in the activity in the motor cortices, there is no difference in the Fano factor.
Right.
So this difference cannot really be explained by having some more noise injected into the motor cortex.
Very nice.
All right. So, I mean, then the question that we had when seeing this increase in dimensionality under HFS was to kind of understand what was the use of having this low dimensional representation in the motor cortex. And the claim that we make is that actually this enables generalization of adaptation. And in order to have a measure for this in the neural activity, we took a generalization performance metric that was first developed in Stefan Ufuzi's lab, and which is called cross-condition generalization principle. And essentially, it consists in training a linear classifier for discriminating between here, for instance, green and orange targets, and take this linear classifier and use it in order to discriminate for the blue and purple targets on the right. So we are trying to generalize the classifier that has been learned on the left targets over there to the one that are on the right side of the circle. So we chose 16 such dichotomies to train that share the same symmetries to train the classifiers on. And this is the difference in cross-condition generalization performance that we saw between control and HFS for each of these 16 dichotomies. And we see that for most of them, the generalization performance goes down under HFS, and likely being due to the fact that this manifold is actually in higher dimension than the one for in the control condition.
Question. So when we have higher dimensions, does that not make it easier to generalize? Because I guess I'm a bit missing the main point here that you say with lower dimensions, generalization is easier.
So here the task, I mean, the physical space happens in 2 dimensions, right? So one could say that a very compressed representation of the task should lie on a plane, right? So essentially here we say that because we're on a plane, if I learn this decoder over there, I'm going to be able to generalize it from the left to the right, right? Because just through this translation, I mean, I recover the same dichotomy between blue and purple. While if green and orange were in a completely orthogonal dimension subspace as blue and purple, then I mean, this decoder wouldn't work anymore. So this means that if I learn something or adapt to something in the case of the left targets over there, I'm very not likely to be able to generalize this to the ones on the right.
Correct.
So this is what this is the measure that we do purely on the neural states. And we just want to probe that a linear decoder has decreased performance when we want to generalize it from one target set to another. And the thing that we also see this time in the behavior is that we kind of recover the same type of observation. So here the way it works, so this is behavioral data, is that as we mentioned before, the monkey is adapting to one target at a time. And in this set of trials, the monkey was surprised by being shown a new target. Right? So it's adapting to target one, and all of a sudden target 3. And so, this is done while a force field is being applied, and we measure how well the monkey performs into adapting to this surprise trial. And essentially, what we see is that during control, well, the monkey has a lot of error in the early trial, so while it has not yet adapted to the first target it was being shown. While for the late trials, under control condition, the monkey was quite able to generalize, which was almost not the case in the HFS condition.
Okay, Hugo, so then this is the plot that I've been pretty much asking for from the beginning of the talk. So you do see a difference in the adaptation when you take the cerebellum out.
In the generalization of adaptation, yes.
I see. So sorry, this is the generalization, meaning that, right, so you introduce a different force field at the end.
Not a different force field, just a different target.
A different target.
And same force field.
I see. And when you say early trials, you mean if this different target was introduced early on compared to late? Because early on, the generalization has not happened in either condition.
Correct.
But later on, supposedly with the reduced dimension in the, yeah, very nice. Beautiful.
All right. So essentially, Elmaleh in Jonathan Kadmon's lab designed.
Sorry, one question. So the
Yes.
generalization, you did it on the 180 degrees opposite. So it's actually like that because is it that? So you did the opposite target or any target?
You mean behaviorally?
Yeah, so when you showed a different target, was that the target whose direction was 180 degrees opposite to the original force field?
So I'm afraid I don't have this information, unfortunately. I don't recall.
Okay, right. Okay.
May I ask a question? Sorry.
Sure.
You are telling that the dimensionality reduces when we actually have cerebellar input, right? So why are you telling that the generalization is related to the increase in the dimensionality? What is not the effect of cerebellum? I mean that the cerebellum is doing the generalization, and when we are impairing that, we cannot see the generalization. It's not because of the increase or decrease in the dimensionality of the neural data in motor cortex or cortex. Because the title of the slide is kind of relating the increase in dimensionality to the decrease in generalization. But shouldn't the increase in dimensionality be impairing cerebellum?
I'm sorry, can you repeat the last bit of your question? So isn't it that the increase in dimensionality?
Is the decrease in generalization due to the impairing of cerebellum or the increase of dimensionality?
So yeah. So here what we show is that essentially this decrease in generalization performance can be explained by the increase in dimensionality, which is in turn, a consequence of not having the cerebellar signals.
I guess what Sana is saying is that one idea is that you take out the cerebellum, that increases the dimension of the population, and that reduces the generalization. But she's saying that what if the cerebellum itself is the one who learns the generalization, and so the reason we don't generalize when the cerebellum is out is that because this component who underlies the generalization is gone.
I agree. I mean, it's probably a combination of both, I would say. I mean, it's. I'm not saying here that the cerebellum doesn't do any bit of the generalization, right? Here again, I mean, we're looking at the preparatory activity,
yeah,
and how this can be linked to generalization already. Yeah.
I guess this is the maybe the key answer. I don't know, Sana, if this is you find this convincing, but this is during preparatory activity, so the animal has no idea what's this, whether they're going to receive a different target or not. So yeah, I don't know.
But it may be that the reduced dimensionality when the cerebellar signals are available actually come from signals that were produced in the cerebellum and already kind of drive the adaptation in the motor cortices, right? But these signals originate in the cerebellum. Then it's hard to say whether it's whether this decrease in dimensionality is a direct consequence of the cerebellum or not. Yeah,
behaviorally, I agree that we don't know, but the neural data that you're showing with the decoder generalizing, that's happening during the preparatory activity. So whatever activity we have in the motor cortex during the preparatory phase, because it's very high dimensional, that activity cannot generalize. So
correct.
Yeah.
Right. So lastly, this is going to be my last slide. Lee in Jonathan Kadmon's lab was able to design a model, the aim of which was to make like the simpler model as possible, where we have this cortical circuit unit here that's essentially a recurrent neural network that's being fed a high dimensional input. So here, essentially one hot encodings for with one dimension corresponding to each of the 8 targets. And then 2 cerebellar units that are going to encode the position of the, what would be the position of the controller in this, in the motor output of our network. And then there is a feedback coming from the cerebellar units, so low dimensional feedback, into the cortical circuit. And what Lee did is that she essentially modulated the feedback gain over there to kind of try and understand how cerebellar feedback could be responsible for this decrease in dimensionality and then related to this cross-condition generalization performance. And so what she saw is that indeed, as you decrease the feedback, that means when you block the cerebellar signals, well, the dimensionality measured in the cortical circuit increases, while the cross-condition generalization performance decreases. And so this essentially goes in the same direction as the data that we had from these motor cortex recordings.
Question, Hugo. So I didn't quite understand what was exactly in the model that managed to do this thing of reducing beta leads to higher D. So how was the model structured? These networks, how were they modeled that they allowed this to happen?
So here you have this recurrent neural network, right? That that has some fixed connectivity. So here the connectivity is not being learned. The only thing that is being learned are the weights projecting from the cortical circuit to the cerebellar unit. And so the network is trained so as to to translate a high dimensional input into a low dimensional one that would be the motor command, right? The motor output. And what Lee was able to show is that as the feedback coming from the cerebellum decreases, the internal representation of that you can extract from this cortical circuit increases in dimensionality.
Okay, so because I'm actually very curious that what kind of a network model can lead to this interesting phenomena that we're seeing, that when we remove the feedback, we actually increase the dimensionality. Because one would have thought it's the opposite, because more signal means higher dimensionality. So question, was this network model trained to recapitulate the behavioral task, or just a simpler, where it has nothing to do with delay execution? Did it have any of those phases?
So it doesn't have delay execution and so on. The model is quite, it's been designed to be as simple as possible. So you don't have the refined preparatory activity, etc., or different signals that are the Q signal and the go signal, or maybe just a short amount of time for which the input is being presented and then nothing, and the network has to execute the movement only within a delay and so on. So you don't have all of this.
Okay, and then without those, then what we have as input is a high dimensional input, and that reflects, for example, the 8 targets.
Yeah. So here in that particular case, the input is in 8 dimensions, right? And the output is in 2 dimensions. Okay. And so we want the output, we train the output to be the correct one, to be matching that of the desired output, the desired target position, and that's it.
Okay, so we have a network that has recurrent neural nets for the cortex, and then it has to learn the connections from those units to your cerebellar unit. And then it also has a feedback from the cerebellum. Yes. And this network is learning to reduce 8 dimensions to 2. Mm-hmm. And then you're saying that when we increase beta, the dimensionality goes down. And I want to know why. What was specific about this network that made that happen?
Well, here the cerebellar output is low dimensional. So you could say that, I mean, the more low dimensional output you feed back into the network that first had a high dimensional input, the lower the dimensionality of the cortical circuit would be. So here, essentially, you're injecting a low dimensional signal into this circuit. So, I mean, you could expect it to have a dimension that would be reduced.
Right. I see. Okay, and then the input goes to cortical units?
Correct, yes.
And the output comes out of the...
Of a projection of the cortical units, from the cortical units.
So the input and output are from the cortex, and then cortex has this loop with the cerebellum.
Yeah.
And then you're saying that when, in all cases that you're showing in your plot, there is a beta, there is some input from the cerebellum to the cortex. However, if this beta has low dimensions,
so
then
it's not about the dimension of the beta here. Beta is just the gain of this feedback, which is always low dimensional.
I see. So when you say beta over alpha, what is that?
Alpha is the gain of the input. So here, I mean, because of the way the network was built, I mean, alpha, the gain of the input that is high dimensional, is of importance for.
I see. So the feedback is always low dimensional, but it's kind of like the strength of that cerebellar feedback over the strength of the input. And so the higher the strength of the cerebellar feedback, then the lower the dimension. Why? Because the cerebellar feedback was low dimension.
Correct.
That makes sense. Then that makes a lot of sense. Right. Okay.
And then, so it was just, I mean, a model in order to probe essentially the correlation between decreased dimensionality and increased cross-condition generalization performance. On the network,
except the only surprise, I'm sorry, the only surprising thing here is that with HFS, you would think that we're stimulating these more. So you would think that we will be pushing this to higher values of beta over alpha, because it's like we're stimulating this pathway more, but it goes in the opposite direction, as if we are removing it.
Yep. So I mean, then I don't know really how
the simulation was done. And I suppose it really depends on the parameters, because maybe the simulation parameters are kind of in a null subspace. It doesn't make it to the cortex.
It's probable. I mean, also, I mean, the high frequency stimulation, essentially, I mean, yes, indeed, brings everything in a subspace that is completely orthogonal to what the cortical areas are used to integrating. So it's probable that in the end, they don't get anything from...
How do we know it's orthogonal?
Because it's unlikely that, I mean, this hand-picked high-frequency stimulation would be mapping directly to the...
Do you actually, when you do it, do you see that the cortical activity goes up in the recording?
So actually, when we do the HFS, we see an overall decrease in activity in the cortical units. So we don't see an increase, rather the opposite. But then, I mean, the precise mechanism of the effect of having this HFS in the fibers mapping from the cerebellum to the thalamus, and then how this has an effect on first the thalamus and then maps onto the cortex. This, I mean, I think so far is yet to be elucidated.
But the fact that the overall activity in the cortex decreases with HFS, with stimulating cerebellar autonomy. So is the whole dimensionality topic not a consequence of that reduced firing rate?
I don't think so. So we essentially kind of correct for this, right? So each of the, so it's something so in the making of these principal component and this matrix, what we do is that we center and also we normalize each of the activity for a given neuron in control and HFS, right? So, as the activity is always centered around 0, the fact that the global activity decreases, is corrected for in the analysis we make.
Okay, thank you.
All right. So, well, this leads us to my conclusion. So essentially, the main takeaways, I would say, from our work is that, well, we studied the effect of blocking cerebellar signals on cortical preparatory activity.
I think we don't have your conclusion slide. We're seeing something else.
Oops.
Yeah, it's not moving. Maybe you do a slideshow again. Somehow it's frozen.
All right. Can you see it now?
I think it's trying to come. For now, it's a blank black screen.
It is fine for me, actually. I guess.
Oh, really? Okay, then I'm sorry. Okay, then go ahead. Go ahead. Maybe it's something about my internet. Go ahead.
And so 3 main takeaways, I would say, from this work is first that the cerebellar signals induce a low-dimensional preparatory activity in the motor cortex, as we saw here and as is depicted here. We also saw that this low-dimensional structure promotes the generalization of adaptation. And finally, we saw that without cerebellar signals, the cortex compensates by re-aiming, but still struggles to generalize. Right. So with this, I'd like to thank you and all of the people who are involved in this work. So this is our preprint for the moment, and the good news is that we just got accepted for publication in Nature Communication, so it should come out quite soon.
Awesome. Such an amazing and inspiring piece of work. Thank you. And you did a great job with answering all these questions that we had. Okay, do we have any other questions?